delighted today to welcome Gordan Lauc, who is visiting us from beautiful Croatia. Dr. Lauc has a PhD degree from the University of Zagreb in Croatia, and has been a pioneer in the study of protein glycation or glycosylation. And he's did a postdoc at Johns Hopkins, and from there went and actually founded his own institute called the Genus Glycoscience Research Institute, which is in Zagreb. He's still affiliated with the University of Zagreb. He's also known in the field for inventing or developing this novel biomarker of aging called Glycan age. And so today, this is what we're going to be hearing about, is this new test and its relationship to aging. So welcome, Gordan. Delighted to have you.
Thank you. So thank you for the introduction and for all of your time to listen what I'm going to do. I'm a little bit jet lagged, so if I say something stupid, it's just the wrong word came out of my mouth. Yeah, just ignore it. So I will tell you something about glycans and how they can be used as biomarkers, but also how they're important as a functional effectors of not only aging, but many other disease processes, which can actually be modified. Not so many people work on glycans. Sorry, just let me turn the pointer. And unfortunately, they're not covered well in any level of education. So I believe you don't know much about glycans. But when we think about glycans, I think one of the best ways to imagine this famous protein, this is the most famous protein at the moment, the S-glycoprotein of the SARS-CoV-2 virus. And all these hairy things on the surface, these are the glycans. And unfortunately, many scientists, including the, at that time, director of NIH, are looking at only the polypeptide part of a protein, which looks like this. And the best analogy to that I can make is studying this poor bird without a single feather. So you can have a well-planned and well-executed experiment on this bird. You can study bird's sight, you can study physiology of a bird, you can study many other processes of this bird, but if you use this bird as a model, you will never understand flight, because bird without its feathers cannot fly. The same thing goes for proteins. Proteins without their glycans, they look similar, but they're lacking some of the key functions which are encoded in glycans. And the majority of proteins are glycosylated. So if we want to understand biology, if we want to understand what is going on in a process that we are studying, we also have to look into glycans. And a word of caution at the beginning, glycation is something completely different. So glycation is a chemical process between the glucose and amino groups on the proteins, which then get converted. You have this advanced glycation end products. We usually quantify it as HbA1c, glycated hemoglobin. Biologically, glycation is like taking a shotgun and firing at the protein, random damage. No meaning in that, just increased glucose, more damage to the protein. Glycosylation is a sophisticated process regulated by multiple genes. And these multiple genes work together to decide which kind of a glycan structure will be attached to the given glycosylation site. And the consequences of alternative glycosylation, so of adding different glycan structures to a given glycosylation site, are the same as consequences of coding mutations. So we know that coding mutations is something bad. Coding mutation can kill you because it's changing the structure of the protein. Alternative glycosylation is doing exactly the same thing. It's changing the chemical structure of a protein, but not by changing the sequence of a single gene, but by changing something in this complex network of genes which regulate the glycosylation of a given protein. And I think it was nearly a decade ago, 2014, we published an opinion perspective article saying that the glycans are the third revolution in evolution. So the first revolution was nucleic acids. Then the second revolution was the invention of proteins. And the third invention was the glycosylation, which enabled the multicellular life. And we didn't prove it, of course, because you cannot prove anything in evolution, but we found the sufficient evidence to claim that this is actually happening by looking at collaborative cross cohort of mice generated by Grant Morahan in Western Australia. So he started with 8 very different founder strains, crossed them for 3 generations, and then inbred them into strain. So he created strains of cousins, nearly clones of cousins, which can be studied. And then when we looked at IgG glycan composition of these mice, some of the strains, so each symbol is a different strain of mice, became really completely different, completely separated from the rest of the mice by only 3 generations of reshuffling allelic variants. So there are no mutations here. It's just reshuffling of allelic variants, which when they're fixed, they generate a chemical structure which is different and which is then functionally different. So these mice are functionally different because they have different glycans attached to immunoglobulins. And it's not only me claiming the glycans are important. They're very strong policy documents, both in Europe for 15, 16 years now. And 10 years ago, the US National Academy is claiming that glycans are directly involved in the pathophysiology of every major disease. And if you want to understand biology, we really have to look at the glycans. And even glycans got the centerfold of the New York Times because of the COVID. And of course, glycans are essential for all aspects of COVID. But glycans are chemically very complicated. So they're nonlinear structures, and different structures can be attached to given glycosylation site, and many proteins have multiple glycosylation sites. So it really requires many chemical steps, complex analytics to be able to see the glycosylation story of a given protein. And at the moment, we can still see only part of the picture. So there are many aspects of many different forms of glycans, like glycosaminoglycans, glycosingolipids, glycoproteins. Glyco-RNA has recently been discovered, which is crazy. I have no idea what is it doing. Nobody knows what it's doing. Free oligosaccharides. Majority of people still focus on glycoproteins. We still cannot look at the glycoprotein as entire entity, but we usually cut it in glycopeptides or release the glycans and then look what can we see. And the technology is rapidly progressing. So 15 years ago, somewhere here, we published the first study on over 1,000 people. Before that, only small cohorts have been studied. And now we routinely do studies on over 10,000 people. So it's really possible now to do reasonably high throughput studies of large cohorts of humans or animals and get the glycan information. As I mentioned, mostly either by releasing the glycans and then studying them either by HPLC, capillary electrophoresis, or MALDI, or cutting the protein into glycopeptides and looking at glycopeptides either by MALDI or LCMS. And my lab analyzed over 150,000 samples already. And actually, out of 190,000 published glycoms, we generated 159,000. So we currently more or less dominate this field. And this is a large chemical review article, which is coming out in actually a couple of weeks, and we got a cover page. And what we are trying to do, and one of the reasons why I'm here, is that we are trying to look into glycans in collaboration with best researchers which are asking the best biological questions. So we are doing it with David Sinclair from Harvard, Nishi from Imperial College, Adam Battenberg from Cambridge, many people from many leading institutions who have good biological questions, good biological samples, and then we provide glycan information which can help them understand what is actually going on, to see the whole picture, not only the bird without the feathers. And in the last couple of years, we really managed to push these glycomics into the, still not the mainstream, but we can publish in a leading journals. We have a couple of gastroenterology papers, circulation research, a couple of Nature series journals, So it is possible to publish good glycan data into good journals today. And one of the proteins we focus a lot is immunoglobulin G. For immunoglobulin G, we know that glycans attached to the Fc domain are essential in regulating effector functions of IgG. So IgG binds to antigen with the variable region, but the constant region activates the effector functions. And normally, the variable region is defined before we know the nature of the antigen. And when the immune system meets the antigen, it has to decide whether it's a virus, which will be inactivated in one way, or a bacteria, which will require another branch of the immune system, or just food, or something what we inhaled and which should be just ignored. And this is being determined by different glycan structures, which are being added to the Fc domain. They change the structure of the Fc domain, and then it binds to the different receptors. There is a very comprehensive review published in this book. Book is horribly expensive, €3,500, but our chapter is freely available. You can download it from here if you are interested. And the field of immunology is starting to realize that. And our previous review, published in 2018, is still the most downloaded and the second most cited paper in cellular immunology. So glycans are coming to the mainstream, especially with this COVID pandemics. People understand how important glycosylation is. As I mentioned, we went that route the first 15, 20 years ago. And one of the first things we learned is that we are all different. This is just a random sample of 6 individuals, different color is a different glycan attached to IgG. And just by looking at it, you can say people are different. And actually, large part of interindividual differences are chemically glycans. Even the blood groups, ABO blood groups, are chemically glycans. And this is regulated in a very complex manner. So even the pathway for biosynthesis is complicated. But what we have shown is that this maybe 300 enzymes which are involved in the story are only tip of the iceberg of glycosylation. So enzymes are shown here in orange. So we have 4 enzymes which are involved in a synthesis of IgG glycans, and one which is here just from other reasons. But there's over 40 genes which regulate IgG glycosylation. So we published a series of GWAS papers in the last decade and are mapping this network of genes which actually make a decision how to glycosylate IgG. Because glycosyl transferases are effectors, they're enzymes which do the work. But somebody has to tell them what to do, and these are these regulatory networks. And these genes, which regulate IgG glycosylation, show very high pleiotropy with the different diseases and conditions. So we have mapped 94 different phenotypes, which share the same SNP, so at the SNP level, for the risk for things we would expect, like lupus or type 1 diabetes or Crohn's disease, where we know antibodies are important. But we see also pleiotropy with the Parkinson's disease, with some types of dementia, with cholesterol levels. So there are many different things where the same genes, which are risk factors for a disease or condition, are in the same time regulating IgG glycosylation. And these networks are protein specific. We did the parallel GWAS of IgG and transferrin. So we had the same 2,000 people isolated in the same time, IgG and transferrin, analyzed them, analyzed their glycans, and did the GWAS. And transferrin and IgG have a very similar glycan structure. It's only these simple biantenary glycans, and the enzymes which make them are the same, but the regulatory networks are completely different. So the regulatory networks is something which makes a decision how to make a glycan structure, which will perform a biological function. And in addition to multiple genes, to multiple variation in the gene sequences, which affect the final glycan structure, there is epigenetic regulation of all of these genes. Because, of course, each of these genes can be either activated or deactivated by epigenetics. And there are some direct environmental factors, including the hormones, the diet, which all integrate into the final glycan structures. So we have genetic polymorphisms, we have epigenetics, kind of a memory, lifetime memory, and then we have a environment which work together to decide what kind of glycan structure will be added to specific glycosylation. And still, this is significantly heritable. So despite such a complexity, glycan composition is between 30 and 70% heritable, different glycans, different heritability. And this is something we have determined by studying a large number of monozygotic and dizygotic twins together with Tim Spector in UK. And the glycan age that I will mention later is 40% heritable. So 40% is heritable part, the other is environment. And we are trying hard to functionally validate all of these GWAS hits we are finding. We have developed a special set of, we call them epi-CRISPR tools. They're based on CRISPR, but the nuclease domain is deactivated, and then different domains were added either to methylate, demethylate, or just activate the gene VPR or inactivate the CRAB, and so on. And this is one example what we did for estrogen. We all know that estrogen is very strong regulator of IgG glycosylation, but estrogen has many different functional consequences. And if we can map the network, maybe we can target a specific gene which is downstream of estrogen, which will affect IgG glycosylation, suppress inflammation without all other consequences of estrogen. And this was published last year in Frontiers of Immunology. And here I have just listed, if you're interested, this is our epi-CRISPR tool. These are all the things we can do. Activate VPR, deactivate, methylate, demethylate, a number of histone modifications which can be added to the given region to study this specific gene to try to understand its role. We do it in IgG glycosylation, but this can also be done in other things. Sure. So I'm a glyco person. So it's actually my wife who is leading this epigenetic part. So some glycan structures act as ligands for specific receptors. Selectins bind and then activate something, but some of them can be considered just as a part of a structure. So, but it's a structure which is not defined by a sequence of nucleotides in a gene, but with the network of genes, which then adds a given glycan, and then the protein is different. And this is why I said the glycosylation is the third revolution evolution, because it enabled structural new structures, meaning new functions, which are not directly encoded in a gene. Because if it's encoded in a gene, you need a mutation to change it. If it is encoded in a network of gene polymorphism, then you can just reshuffle the polymorphisms or even epigenetically reshuffle activity of genes and create a novel structure. And I think this is one of the key mechanisms which actually enables us to compete with all these bacteria and viruses, because they mutate like crazy and we still can fight them. So let's go to aging. First, we thought of it as a problem because we were studying diseases. And we realized that if we don't have a perfectly matched controls, age-matched controls, our data was useless because the effect of aging was stronger than the effect of the majority of diseases. So here, when you look, this is 5 different large cohorts. Blue are men, red are women, different glycan structures attached to IgG. They change with age. In men, it's mostly linearly. In women, there's this strange acceleration around the menopause. I will come later to that. And this was the basis for this glycan age test, which we developed. And of course, here I have to disclose, I'm heavily conflicted. I'm an inventor, I'm a co-founder of a company. So I do have clear commercial interests here. So you have to evaluate the evidence I'm showing yourself. But what we know is that people when are young, they have this type of glycan structures, which are generally suppressing inflammation. And as we are getting older, and this change starts to accelerate in the mid to late 30s. So actually, this is the time when there is no more genetic pressure to keep us alive. And then we become more and more pro-inflammatory. As we are older, we have most of these pro-inflammatory glycans. So we constructed this glycan age clock, which is, at the time we published it, and it was actually published in the very same day as the epigenetic clock, the fifth original epigenetic clock. We were guessing the chronological age plus minus 9 years. At that time, it sounded good, but of course, methylation is way better in predicting chronological age. But besides potential forensic application, we have a drop of blood or person claiming they're minor, you don't really know, you don't need the biomarker to see somebody's chronological age. What is interesting with the glycan age is that the delta, the difference between chronological and a glycan age, strongly correlates with unhealthy lifestyle. So things like insulin, fibrinogen, HbA1c, BMI, triglycerides, glucose, weight preferences, these are the biomarkers which strongly correlate with the delta glycan age compared to chronological age. And when we look in the different parts of the world, and we compare 27 different populations from Papua New Guinea to European cohorts, people are different. So people living in the different parts of the world are aging in a different pace. And actually, the thing which correlates the most is the expected lifespan. So if you live in the countries like Uganda or Papua New Guinea, your glycans will move much faster to this old glycan than you live in the developed West. If you are in the middle developed countries, you're somewhere in the middle. So environment is something very important.
I'm thinking my analogy to the methylation clock that I was more about. Are these differences, are there different location clocks or interesting implementations, or is that the implementation is kind of a single consistent set of changes in age?
So this is the same type of change. It just happens faster in people who live in areas where their expected lifespan is shorter. I'm not trying to dissect genetics from environment because we don't have hard data yet, but we have some indication, and this was done in a cohort of people who moved to London from either Afro-Caribbean region or India. And the second generation immigrants were more or less the same as the white Caucasians. So partly it's genetic, but big part is where you actually live. And the question about epigenetics, acceleration in glycan age and epigenetic and age do not correlate much. So this was published by a group in Edinburgh. We were not involved in that study. So they were comparing acceleration in the different epigenetic ages and all other many different aging clocks. And when you look at correlation between epigenetic age and glycan age, acceleration in, because the age itself will correlate highly because they correlate with the chronological age. But when you look at acceleration, the acceleration does not seem to correlate. This is just one paper, one study. Maybe it will not be replicated. But for some other clocks, there was much higher correlation for glycans and epigenetics was not. And I'm not too surprised because epigenetics behind the glycan age clock is in bone marrow, is in plasma cells in bone marrow. So we don't see. Epigenetics people are measuring is usually from the blood, where there are different blood cells which do not directly affect IgG glycosylation. So I think epigenetic aging is more about the intracellular aging, so the cell aging, while the glycan aging is more about the organism aging and the immune system changing with age. Another very, so another very important aspect of changes in IgG glycan, including the glycan age, are different diseases. In our 150,000 people we analyzed, there were dozens of cohorts with people with different diseases. And what you can see here are different glycans, so G0, G1, G2, whatever they mean. The last four bars is aging. So comparing older and younger people in 4 different cohorts, and all other bars are diseases. And here we have a couple of lupus cohorts, ulcerative colitis, Crohn's disease, rheumatoid arthritis, multiple sclerosis, GVHD, type 2 diabetes, colorectal cancer, sepsis, HIV. Most of these diseases, especially in glycans, which are glycan age related, the G0, G2, and S, they change exactly in the same way as aging. So people with the disease have a glycom with similar composition as older healthy people. And in most of the cases where we looked, these glycans change up to a decade before disease is being diagnosed. Because disease is an endpoint. And to come to the endpoint, there has to be a continuum of molecular changes. And we believe glycans are part of it because they're functional effectors which regulate inflammation. And in most of these diseases, inflammation is one of the important components. So what I think we see here, we see a molecular pathology, which is increased pro-inflammatory potential of IgG, which then manifests as some of the different diseases, depending on a genetic, epigenetic, environmental factors in a given person. Because remember, disease name is something invented usually over 100 years ago, based on a diagnostic criteria available then, meaning you have inflammation in joints, arthritis. You have pain in your stomach, it's IBD or whatever. Albumin is not glycosylated. No, albumin is not. So it's enzymatic glycosylation. Yeah. So IgG has a bit different glycosylation than the majority of plasma proteins because I have structures here. So what IgG usually has this core fucose, which majority of plasma proteins could not have. And it has only biantennary glycans, where the many plasma proteins will also have the larger glycan structures. So the liver does make a little bit different proteins than does the B cell for the IgG. But the B cell also makes a different protein, different glycans for other proteins. IgG is something special. I think IgG glycosylation is a very sophisticated tool developed to control our immune system. They direct, so some of them direct IgG to different Fc receptors. For example, the core fucose is a safety switch, which is preventing binding to the Fc gamma receptor 3A, which is activating ADCC. So it's the killing the target cell. So majority of our antibodies are not supposed to kill the target cell, so they put the core fucose not to activate it. If, for example, we have a viral infection like COVID, then we want to make antibodies without the core fucose, because the antibodies are not attacking the virus, the antibodies are attacking the cell which is infected. So then it binds to the cell, activates NK cells to the Fc gamma receptor 3A and kills the target. Some other aspects are binding to the different receptors. Some glycans might even not bind directly to the receptors, but compete with the cell-cell interactions like activation of eosinophils and so on. Many different functions. So the most famous disease, COVID. David Sinclair and I speculated in spring 2020 that it will actually be the biological age. It will be a better predictor of disease severity than the ecological age. And indeed, we have shown that when we looked at the first cohort, this was the first wave cohort, we do see the consistent change even after correction for aging, that people with a higher biological age had a more severe disease. But interestingly, people who had a more severe disease also had accelerated aging in that period. So if the COVID was mild or asymptomatic, IgG glycom did not change. But if people had severe COVID, and in particular those people who eventually died, their IgG glycom changed rapidly in a matter of days, actually 7 to 10 days. These people would age 5, 10, 15 years. Even I aged 5 years when I had my first COVID, and it was not really a severe form of COVID. And so definitely different people will generate viruses with different glycosylation. So as far as I know, nobody yet did a proper population study because it's not easy. You have to actually isolate the virus from a patient. You cannot cultivate it because then the glycosylation changes. But what we are currently trying to do, we are trying to look whether inter-individual differences in glycosylation were kind of a barrier for transmission. So we are looking for people who were exposed to the virus but did not get infected, whether it was actually glycosylation difference between the pairs, which also prevented the transmission. Because what we know that with the different variants of a virus, some people who were initially resistant got infected with the second one. Some were claiming, you know, I can't get it, and then the Omicron came and they got it, and so on. So glycans do play a very important role there. But it's not only COVID. So the COVID is not that special virus. Influenza does a very similar thing. So if you have a severe influenza, your IgG glycan will also change rapidly. This is still unpublished. And we have another paper which is coming soon. And this is when you have pneumonia, if it's a viral or bacterial, glycosylation will change in a different way. So our immune system is somehow recognizing it's a viral or it's a bacterial pathogen and tuning the glycosylation in a way to kill that pathogen.
Do people with diabetes
So glycosylation changes in diabetes. Not so much IgG, but some other aspects of glycosylation, which I will show later. We haven't looked at the specific differences between diabetic and non-diabetic people who got COVID, because we were really trying hard in our COVID cohorts to eliminate all other comorbidities. We wanted to see the effect of a virus. So our paper, actually the paper is coming out on Monday, 27th in eBiomedicine. We started with 1,000 people who were hospitalized, and we included at the end, I think, only 80 of them, because all others have very severe comorbidities, which we didn't want to have here in the story. I will mention diabetes later, but for COVID, we don't know. Everything would be interesting to look at. One of the reasons I'm here is that we can analyze 40,000 to 50,000 samples a year. We don't have that many interesting samples. So if you have some interesting stories, we can get in touch. Maybe we'll set up also something here to make things easier for you to add this glycan information to whatever you're doing. This is also an important question. So did diabetics who get COVID were different from others? Don't know at the moment. So one of the biggest questions we have is, are these changes a cause or a consequence? And I think they're both. So in some cases, it will be just a consequence, but in many cases, it will be also a cause of it. And there are many papers suggesting that glycans change much earlier. These are actually early papers, over 10 years ago. And what we looked at is, if you remember, glycan aging strongly associates with the expected lifespan. So people whose glycans age faster are expected to live short. And if we are talking about mortality, then cardiovascular diseases are the first killer. So this is where we would expect to see the difference if glycans have something to do with it. So we looked in our large cohort. This was done together with Tim Spector and Jim Wilson from UK. We had around 3,500 people, and IgG glycan composition strongly correlated with the cardiovascular disease risk score. So the risk of future cardiovascular events. But this was just correlation at that moment. But using that data, we managed to persuade people from the EPIC cohort And the EPIC cohort was collected in Europe around 30 years ago. Samples were collected, and then there was a clinical follow-up for a long time. And in that cohort, there was around 500 people who either developed a heart attack or stroke after sample collection. And when we looked at the IgG glycom just plus age, it was a little bit less predictive than the entire AHA score in men, and even more predictive than the AHA score in women. So all the information with AHA score includes to predict cardiovascular events, glycans on IgG have it encoded in it. And there are some evidence it's actually causative. So this was done on mice. We didn't do it, it's independent studies, which suggested that this so-called young IgG, salivary IgG, is promoting release of nitric oxide from endothelial cells to the Fc gamma receptor 2B, while the old IgG glycom is blocking this release of nitric oxide. And when we looked in humans, we looked at 2,000 twins in 3 time points across 15 to 20 years. We see that changes in IgG glycom appear before people develop hypertension. So it does seem to be predictive of hypertension. And in mice, we were even able to show that we can prevent hypertension in obese mice by feeding mice with the MANAC. MANAC is N-acetylmonosamine. It's a precursor of salicylic acid, because what happens with the mice when you feed them high-fat diet, of course, they become obese, but they also lose IgG cellulation. So their IgG glycans age faster. The same thing what we see in humans. So as humans are becoming obese, they're losing IgG cellulation. And in this study, Phil Scholl from the UT Southwestern was feeding mice with N-acetylmonosamine. And these mice, of course, did become obese, but they did not lose IgG cellulation. So this is mice who were on placebo. These are the mice who were given the N-acetylmonosamine. So their cellulation was saved. They, of course, did become obese, but they did not develop hypertension. So this is a potentially extremely interesting. Unfortunately, nobody did the study in humans yet. There was a small study showing that it's actually safe. So I hope one day somebody will do a proper trial in humans to see whether also we can protect obese humans from hypertension with a simple dietary supplement. Diabetes is another story where we believe glycans are really causative, because there are several lines of evidence showing that they, in addition to affecting the insulin resistance, they're also affecting the half-life of the insulin receptors, glucose receptors, because all these proteins are glycosylated, and different glycosylation affects their half-life on the membrane. So they seem to be important for glucose regulation. And then we again went to that same cohort of EPIC people collected 30 years ago. They had 70 and 43 cases of diabetes. We divided them into discovery and validation group, looked at the predictive power and the composition, but this is not IgG glycom, this is the total plasma glycom. So glycosylation of all plasma proteins was as predictive, or even a little bit more predictive, than the entire German diabetes risk score. And here, it's not predisposition, it's a kind of continuous process which happens as people are approaching the diagnosis. So again, the same 2,000 twins in 3 time points in 20 years. Some of them developed diabetes. Well, actually, this was a combination of diabetes and insulin resistance. So this is diabetes and insulin resistance. This is 2 to 4 years before, 5 to 7, 8 to 10 years before you develop insulin resistance. So glycans start changing at least 7 years before people develop insulin resistance, which seem to be partly causative for diabetes. And it's also predictive of both prevalent and incident diabetic complications. And we know this can be reversed with the beneficial lifestyle interventions. So people who are losing weight, they will change in this opposite way as people who are going towards it. we still do not know whether this is causative. We know that beneficial lifestyle intervention will decrease the risk of disease, but we don't know whether glycans are instrumental there. There has to be a lot of functional studies to prove that. And another question, the last part of my talk is, can we reverse it? So what can we do to change our glycan clock of aging? And can we make a decision to change it and actually slow down or stop inflammation by doing that? So Glycan Age is commercially available for nearly 2 years now. Many clinics use it, and Joseph Raphael in New York was one of the first. He already tested, I think, over 200 people. And majority of his patients who are in clinic for some time would be 20, 30 years younger than their chronological age. And usually when he takes somebody in a praxis, they're not that better, but with time, they become better. So this is just incidental. This is not the science. I cannot say this is actually working, but this is something what we are seeing. And we're also seeing it in some people we're tracking for a long time. For example, Tim Spector, he's a famous epidemiologist in London, and he was reasonably well for a long time. And then he had a difficult period around the pandemic story. He got worse. And then he decided to do something. And he actually lost 11 glycan years in less than a year. And he and I recorded Instagram Live. So if you want to know what he did, you can have a look. Another very important period where glycans change a lot is the menopause. So when we were looking at the population level, we saw that there is this acceleration of average glycan age, because this is the average person in a population, around the menopause age. And we saw it in many different cohorts. And Glycan Age as a company was seeing clients who would be very good for quite some time and then rapidly accelerate. So this specific lady aged over 30 years within a year. And of course, in that situation, people freak out because glycans usually change before any other symptoms of perimenopause. And what we are now trying to position it as a biomarker which can early identify perimenopause, because with a proper hormone optimization, these things can improve. And we got access to a very interesting cohort of samples. This was done nearly, I think, 30 years ago, where the menopause was induced chemically. So the production of gonadal hormones was blocked. And then part of the group, 21 women got placebo, and 15 of them got estrogen supplementation. And women on placebo aged on average 9 years in 6 months, while the estrogen supplementation group did not. So estrogen seems to be very important in regulation of IgG glycosylation.
This is all super cool, but that really is cut, because the last slide was the hormone optimization that lowers glycation rate. If testosterone and estrogen and so forth are having large effects of the glycosylation of women, if you just measure it usually hormones, you know that does not relate, and that's not true. It can help over the long term. So are we sure that it can be reflect more fundamental processes?
We don't know. So we know that hormones are important. They're not the only ones which are important. Yeah. Because the things, glycans can change a lot even before the hormones start changing. So there are people who get much older before. Now, what is the fundamental aging process? I think we still don't know what is really aging. This is definitely not covering all aspects of aging. So Glycan Age is more or less covering aging of the immune system. Some other aspects of glycosylation are also associated with aging. So we have, for example, something which is coming out relatively soon. It's something called meta-age, which is metabolic age, which is more related to metabolic process. So I'm not claiming Glycan Age is the universal marker of aging.
What can you control for hormonal status in creating the glycation clock that you know you're not incidental changes in hormones?
So it takes a long time for glycans to change. So if there is just a rapid fluctuation, we will not see that. It takes a long time to change all the IgG glycans because they have half-life of a couple of weeks. I don't know what we would get by controlling for hormones, because hormones are also important elements of aging. So losing testosterone, losing estrogen has multiple consequences, which are working at the level of organism. I agree, this would not be on an intracellular level. So I think it is very important to be aware of that there are multiple aspects of aging, and that the different molecular clocks or different biomarkers will measure different things. As I've shown before, accelerated epigenetic aging, accelerated glycan aging do not correlate. There are two different aspects of aging. And I believe there will be other aspects of aging, and different aspects will be important for different things. So these things that we are looking are highly related to low-grade chronic inflammation and the development of cardiometabolic diseases, inflammatory diseases, and other problems which come with old age. And this is just one aspect of aging. So I don't see the benefit of controlling for hormones because they're part of this whole process.
Sure, but in the form of fat, well, I wouldn't say, so we know very well that trying to replace testosterone, replace estrogen, favorably, that's controversial and does not include long-term health outcomes. So that's the context for the process that supplementing testosterone makes one 30 years younger. That's not reflecting the long-term health benefits.
I agree.
But like you said, that's not part of the story.
Sorry. And what we see, for example, is that not everybody will respond to estrogen. So we have some women who are really strongly responding to estrogen supplementation, some are not. So there are multiple elements of this entire change. As I said, first, 40% is genetic. So 40% is kind of hardwired, you can't change it. There are many epigenetic factors which act on all these genes, which we don't really understand how they are being regulated. There are environmental factors, so the hormones would come at this environmental level. And in some people, it will be more important, some people it will be less important. And for example, here, if you look, this average, but when you look at some individuals, you change very little. Some of them change a lot. So these box plots can be kind of deceiving, to see. We have to separate them because they're very different. And actually, our first paper saw the difference only in women. So these glycan structures and prediction of cardiovascular events only in women. We have now a replication study, which is seeing also in men a little bit smaller effect. So definitely for cardiovascular events, we have to stratify men and women. I think in everything what we do, we should be stratifying men and women. I think it's wrong to combine us. They're very different. So I have a few more slides and then we can continue discussing. So what we are currently also trying to develop is this, we call it MENAGE now. It's a predictor of early perimenopause. Because I'm a man, I'm not aware of all the problems which happen around the perimenopause, but women are. And we as men are actually heavily neglecting women in all the research. Only recently the women have been started to do research more, which I think is a big mistake. And I think it's important to understand. I think it's important to appreciate that we are different and not to claim that we are all the same. So women should be studied separately from men. And one thing where definitely they're different in these changes of estrogen, which happen around perimenopause. And the problem is that these hormone changes are very uninformative because they go up and down on a daily basis. So if you want to have a reliable measure of change in the hormones in the perimenopause period, you actually have to sample daily and take an average. IgG is doing that for us. So IgG is integrating information about the daily concentration of estrogen. And if you look at the same person, everything else is controlled. So only the change in estrogen is what we see as a change. And this seems to be very predictive for perimenopause. Another thing where we see very strong effect is the weight loss. For example, people undergoing bariatric surgery can have very dramatic changes. We had a patient who lost 37 Glycan years in 6 months. Of course, bariatric surgery is probably too aggressive for the majority of people, but the regular weight loss is also working. These are the 2000 twins we were tracking for 20 years. And those who were gaining weight were actually aging faster than those who were losing weight. We had 1,000 people in Europe in 7 or 8 centers on a low-calorie diet for 2 months. And a low-calorie diet improved glycom in more or less all of these cohorts. But nobody can do caloric restriction for a long time. You know? Nobody wants to be hungry. So the study was designed, it was a diogenes study. They wanted to look which of the maintenance diets were better. So they had 5 different diets. And on each of the 5 different diets, some people got better, some people got worse. And it did not correlate much with the BMI. So it is, we're all different. There's no standard human, and different diets are optimal for different individuals. The same goes for exercise. So we were all taught that you can sweat out the bad diet. It's false. You cannot go into the gym and just compensate for all the bad diets that you eat. And we have seen, especially in the middle-aged people, if they just start exercising, majority of them is overtrained and they got worse. And only the really carefully optimized exercise regimes show very good effects relatively quickly. Another extremely important thing is the microbiome. So glycans and microbes talk in both directions. First, the mucins are feeding the microbes and selecting the ones which we like. And second, the microbes are changing the glycom. We have a couple of papers showing that by fecal microbiota transplantation, you can actually change also the glycom. So to slowly come to an end, a few more. So we have this test, which is, as I mentioned, commercially available. People are buying it all the time, which I think the best advantage of it is that we know what we are measuring. We are measuring molecules for which we know what is the function. And we actually quantify the limited number of molecules which can be reliably quantified. And we know they're on this pathway of disease development. So because disease is a point of no return, usually people go to hospital when it's too late. You can't make them healthy again. You can just alleviate the symptoms. But if you find them here, when only molecules are changing, and this is probably a decade-long process, then preventive interventions can solve the problem. And you know, we all understand that our car needs regular maintenance, that we are checking the tires, changing the tires, changing the oil, and we don't think our bodies need the same thing. And just imagine that if we would change our tires only once they explode on the highway, how would the highways function? And this is what we do to our life, to our bodies. We try to fix them only when they're broken. So I think we need to think more about preventive maintenance. And I think tests like this and other tests, of course, can help, because one of the key things is we need motivation. And the motivation that we will not develop disease in 30 years is not working. You know, none of you will change anything today for a feedback which is coming in 30 years. So we need a faster feedback. And these type of tests can tell us in a few months what is actually going on, going up, going down, and then we can change and correct, because we get this objective insight into what is going on, what we cannot see in the mirror. And at the end, I have to acknowledge that this has been generously funded with a number of European, US projects. We got over 20 million euros, which we burned on glycan analysis for all this source. And I thank you very much for your attention. So heritable change by the definition should not be changeable. This is something what we inherit. But we don't know which component is heritable, which is not. So, heritable is kind of setting your starting point, but then the change is mostly the lifestyle. So this is why I always, for example, my glycan age is horrible, 78. And I don't worry myself too much if I manage to move it down a little bit. I think it's much more concerning if you are 30 and you move to 50, than me thinking at, okay, it's horrible, but I'm not the best example. So I think heritable part we should forget now, because this is something you cannot change. What you can change is diet, exercise, sleep, stress, but you cannot change all four of them. No, not British Queen can do it, but we cannot work on all four fields. So I think this can help us identify what are the most important things for each of us. Unfortunately, for me, it's diet, and I like to eat, so I'm hopeless.
So, you mentioned a little red triangle.
That's the core few codes.
So that's one that,
This is galactose. This is wrong. No, the few codes does not disappear. You're right. The figure is wrong. We have to put another few codes here. So what is mostly happening is the salic acids are being lost, and galactose is being lost. And these molecules are responsible for the suppression of inflammation. For example, the IVIG treatment, when you put immunoglobulins from more or less young and healthy people to the old people, it's suppressing inflammation. And now we know that the salivation is an important component of that. Now pharma companies are trying to put more salic acid on immunoglobulins to make it more efficient.
so the question is can you actually the lighter interaction of the I don't really know how these things work.
Yeah, so
the galactose, for example, to actually increase the consumption of that, which we want to do.
So the problem here is that there are so many different proteins which are glycosylated. And then if you increase salivation of all of them, and by diet, it will not be IgG, it will be many others, we don't know what will be the consequences.
So
No, it's IgG specific. But the experiment we did in mouse with this manosamine and acetylmanosamine, it did improve salic acid. And there is an NIH study on, I think, only 20 people, that if you give young and healthy people this manosamine, it's okay. They don't have consequences. Now there are clinical trials for some of the glycosylation deficiency diseases, trying to improve it with this MANAC. I think what we need obese people taking MANAC to see whether it helps. But it's, as I mentioned when we chatted, I'm very cautious about supplements because we are so finely tuned machines. Then if you push something in one direction, you never know what will happen.
Well, basically, changes over time.
So we know, so it's not removal. This thing is not removing. It's not removal. It's all most of it is de novo synthesis. And we have the map of genes. I'm just looking back at it. So as I mentioned at the very beginning, so we did this series of GWAS papers, and currently with unpublished hits, we have over 40 genes, which associate with the different aspects. So these here, actually the green one is one of our first kind of targets, because ELL2 is associated stronger with salivation, but there are some others which are also involved. So as I mentioned, aging, there are many different aspects of aging. So say genes associated with aging, maybe they don't associate with this aspect of aging, which is more or less aging of the immune system. And I also think it's only happening if we have interaction with the environment. For example, mice, since mice were, since recently, maybe 20 years, they're being, we have this specific pathogen-free facilities. They don't show this change that much anymore. Yeah. So the initial studies were showing this type of change. Now we did several of them, we are not seeing it. So either the mice change, which is also possible, or we would have to grow them in interaction with the environment to have this change of the immune system. Because this change could also be driven with the continuous contacts with the different antigens, which are driving the immune system. Don't know.
So if it's the fight regulated network, then there's a change in the levels of some building blocks. So like then is it that it is a network that the outcome will be different? Or it's well regulated?
So we looked at this small NIH cohort, and in young people, MANAC did not change glycosylation. So the regulatory network was holding. I don't know what happens if the regulatory network is already perturbed as in obese people. So maybe if the network is perturbed, MANAC can actually save it. But you are right. So young people, when you just put MANAC, it did not affect the cellulation because they already had this young level.
One more question. And you showed that there was a dramatic difference when you applied MANAC, but was it like translated functionally? Protection from cardiovascular?
So mice did not develop hypertension. This was the endpoint for that study. So I don't know whether this translates in heart attacks in mice. I really don't know. But so the endpoint was hypertension, and mice did not develop hypertension.
One last question. If you take glycosylation on collagen,
So collagen has this glycation thing, which is a chemical reaction with glucose. It's not glycosylation. I think pre-collagen does have some glycan structures, but the collagen itself, when it's in all these tight fibers, it's not glycosylated. So it might have this glycation, which happens with aging, but this is a chemical damage. This is a different thing.
Okay. I want to thank you, Dr. Lauc, for the seminar, for entertaining us and interesting work. Thank you again. I hope we get a chance to work together.
Thank you for the invitation.